Now, in the last session, we were talking about the structure and organization of a typical prokaryotic cell. And prokaryotic cell here means a cell without a typical nucleus or even with internal membrane-bound organelles. And that is very characteristic for prokaryotic cells. So here again is the image of a prokaryotic cell, a drawing. We have here the cell membrane, which is a lipid bilayer membrane which keeps sort of the cytoplasm, that's the inside of the cell, separated from the environment. And then in this cytoplasm there are a lot of things, components that are important for the survival of the cell. So for example, we can distinguish ribosomes, which are important for protein synthesis. We do have the genetic material inside this cytoplasm here. That's the genetic material. And usually this is DNA. But in contrast to eukaryotic cells, in a prokaryotic cell, there is no membrane around it. And the genetic material is not organized in a nucleus. That's why it's called pronucleus. Instead, we are talking about a nucleoid, that is the region where the genetic material is located in prokaryotic cells. Okay, so what else do we have? Most prokaryotic cells also have a cell wall. That's this blue one here. And this cell wall is made up of proteins and sugars. And we refer to this cell wall frequently as the peptidoglycan layer. Peptidoglycan layer. That is the typical cell wall, and it depends on the prokaryotic cells on the composition of this peptidoglycan layer. Many bacteria or prokaryotes also have a capsule, which is made of slimy stuff, sugars, and things like that, and they contribute to what very often is referred to as a biofilm. This is a sticky substance in which the bacteria are embedded in, and that gives them additional protection from the environment. So for hospitals, this is a big problem, these biofilms, because the bacteria are just clinging to the surface and are difficult to to eliminate. So often we find also in typical prokaryotes some hair-like structures. They are made of proteins and they are referred to as pili or fimbri. And they are, as I said, are made of proteins, and they very often help the bacteria in the attachment to a surface or in the case of a sex pili, for reproduction. So sex pili can be used almost like a penis by one prokaryote to transfer genetic material into another receptive prokaryote of the same species. And we also find in some bacteria, but not all, these longish protein protrusions. They are called flagellae, and they are made of the protein flagellin. And these flagellae are almost like sort of little drivers. They give the bacteria, the prokaryotic cell, the ability to move and to swim. But not all prokaryotic cells have these flagellae. Some have only one. Other prokaryotes might have quite a few of these flagellae, and they help the bacteria to move around. So that was the typical structure of a prokaryotic cell. Now let's have a look at a eukaryotic cell. So let's look at what eukaryotic cells look like. And we can distinguish between animal cells. Let's start with a typical animal cell first. And like the prokaryotic cells, we have a cell membrane. So this here is the cell membrane. Which is made up of phospholipids, and we will discuss that in more detail in subsequent lectures. We also have the cytoplasm, or sometimes are also referred to as cytosol. That is the sort of liquid environment inside the cell, separated from the environment by the cell membrane. We have ribosomes, which are important for the synthesis of proteins. So here we've got ribosomes. They are incredibly complex machineries that help the cell to produce proteins. And of course, we have the genetic material, but this time it is enclosed in a nucleus, in a membrane system. So let's draw it like that. So this would be the nucleus. And it contains the genetic material, the DNA, and for typical eukaryotic cells, DNA is the genetic material that contains the blueprint of life. The nucleus has a nuclear envelope, nuclear membrane, and this membrane actually is made up of phospholipids again, similar to the cell membrane, and it provides a specialized compartment, actually, this nucleus, where the genetic information, the DNA, is stored. But and here we see a very characteristic difference to the prokaryotic cells. Here we have this nicely formed nucleus. Of course, we also have other membrane systems inside this animal cell. So, for example, we have what is generally referred to as the powerhouse of the cell. We have mitochondria. And these mitochondria generate energy and are involved in a lot of biochemical reactions. Again, we have membranes, and in the case of mitochondria, we have two membranes made up of phospholipids, which provide a nice compartment for the cell in which specialized processes are taking place. We also have a very extensive membrane system in the cell, which might look a little bit like this. This is referred to as the endoplasmic reticulum. It forms a sort of a network, and that is what the word reticulum actually means. It is made up of membranes again, and here you see the pattern here. Lipid membranes or phospholipid membranes. And the endoplasmic reticulum comes in basically two forms. One version is studded with ribosomes, and I'll draw it like that. This here is what is called the rough ER. ER stands for endoplasmic reticulum. And the appearance of this is fairly rough, and it's not in a way smooth membrane, because these little dots are actually ribosomes that are attached to the endoplasmic reticulum, to the rough endoplasmic reticulum. And the rough endoplasmic reticulum is involved in the synthesis of special proteins, of secretory proteins. So these are proteins that are transported out of the cell. And it's also involved in quality control of proteins and things like that. We also have a compartment in the endoplasmic reticulum, this one here, which is referred to as the smooth endoplasmic reticulum. And this is predominantly involved in lipid synthesis. Lipid synthesis and calcium storage. And there are no ribosomes attached to the membrane of the ER, and that is why it looks fairly smooth. We also have a system that looks a little bit like sort of a stack of membranes. Something like that. This is called the Golgi apparatus. Golgi apparatus. Again, we've got membranes, phospholipid membranes, forming these stacks, and the Golgi apparatus is responsible for the control, modification, and sorting of proteins. So it basically tells the proteins where they need to go. So for example, they might send some proteins to the surface, to the cell membrane, or when they are secreted, they might be released. And what we find very often are small transport vesicles. These are transport vesicles, membrane-bound compartments that transport vesicles or transport material to certain destinations in the cell. We also find other compartments which have membranes. So, for example, we have compartments, they are called peroxisomes. These peroxisomes have, again, a lipid bilayer membrane. Peroxisomes are typically involved in the degradation of some proteins or cellular components, and detoxification and degradation. So there are some specialized biochemical processes that are happening in these peroxisomes, mainly responsible for the degradation of waste products, however, are other vesicles like that. These are referred to as the lysosomes. Again, they have a lipid bilayer, and they are places that is almost sort of the trash bin of the cell. Everything that needs to be recycled or we can't use really will be broken down in these lysosomes. We also have elements that give the cell its typical structure. So these are long, usually filaments. They are protein filaments. And they are part of the cytoskeleton. Now, the cytoskeleton is made up of a number of these protein components, of these protein filaments. They come in different shapes and forms, and they are almost like tent poles. They can give the cell its typical structure, but they can also be used as sort of almost like railway tracks. And we very often find little vesicles being attached to the cytoskeleton, and they can move along the cytoskeleton. So these are principally the components of an animal cell, of a eukaryotic cell. And the important thing is that we've got lots of membrane-bound organelles in the cell, and very prominently we've got the nicely formed nucleus here that contains the genetic material. One question is, do all cells actually have a nucleus? All eukaryotic cells? And the answer is no, not all eukaryotic cells contain a nucleus, because red blood cells in humans, so red blood cells in mammals, I should say, do not contain a nucleus. Okay. So would we refer to them as prokaryotic cells? The answer is, again, no. We would not refer to red blood cells as prokaryotic cells, simply because when the red blood cells starts off its life in the bone marrow, it is formed with a true nucleus. But when the red blood cell matures, it loses basically all of the membrane components, including the nucleus, and the red blood cell changes its function. It does not have any membrane-bound components, no mitochondria, no endoplasmic reticulum, no nucleus. It is just simply a bag full of hemoglobin that is transported around in the body. So that is for mammalian cells. And because at one point in their life, these red blood cells had a nucleus, they are still true eukaryotic cells. Birds, reptiles, amphibians do have red blood cells, but they still contain a nucleus. So from a forensic science point of view, you can very nicely distinguish whether the blood comes from a mammalian victim or from, let's say, a bird. So that's very important to remember. Okay, so that was a typical animal cell. Now let's have a look at what a plant cell looks like. Plant cells are quite specialized. Let me draw it like that. And again, we've got a characteristic cell wall, a cell membrane, which is made up of phospholipids. And these phospholipids are synthesized and produce this nice cell membrane that keeps the environment out from the cytoplasm here. Or the cytosol. And we, of course, have ribosomes for protein synthesis. Like this. We do have a nucleus, again, because plant cells are eukaryotic cells. So here is our nucleus with a membrane, phospholipid membrane, and it contains the genetic material. So here we've got the DNA. And again, that is pretty similar to animal cells. Interestingly enough, and I forgot to mention that, for the animal cells, we also have a protein structure here. Let's go back to our animal cell. This is a protein structure here. This is called the centrioles. This protein structure, which is important for the cell division. In animal cells, but we don't have centrioles in plant cells. So again, this is quite interesting for forensic science. If you want to, for example, check out whether some food is purely vegetarian, you check whether the cells contain centrioles. If they do contain centrioles, then they are probably animal cells, whereas plant cells don't have centrioles, so no centrioles. Okay, what else do we have in a plant cell? Of course, we've got mitochondria, the powerhouse of the cell with its membrane system. We've got the endoplasmic reticulum. And we can have smooth as well as the rough endoplasmic reticulum. So endoplasmic reticulum. We also have the Golgi apparatus in these plant cells. So Golgi apparatus. We have peroxisomes. for the detoxification and breakdown. Plant cells also contain a structure that allows them to perform photosynthesis. So we have chloroplasts, well, at least green plants. So we have chloroplasts for synthesis of sugars from sunlight. Again, we've got a number of phospholipid membranes in these chloroplasts. And we also have something very typical for a plant cell. And that is actually a huge liquid-filled membrane system. It is called the vacuole. And it has, again, a cell membrane, a wall, I shouldn't say cell wall, a lipid, phospholipid membrane. In the case of the vacuole, this is referred to as the tonoblast. And the vacuole is basically a storage compartment, and also where some cells processes take place. It is the functional equivalent to lysosomes. So usually plants don't have lysosomes, but instead they have these huge vacuoles. And the vacuole will squeeze most of the other organelles to the wall. That's why I've drawn it like that. The plant cell can also have cytoskeleton, something like that. So that's the cytoskeleton. And very characteristic for many plant cells, not all of them, but for loads of plant cells, is that they produce a thick layer outside the cell membrane, something like that. This is referred to as the cell wall. The cell wall is usually made up of sugar, of cellulose, and very often it also contains other components, like, for example, lignin or something like that. And this is a fairly rigid structure that gives the plant cells their typical structure, if you like. And it makes it quite difficult, actually, for things to get into the cell. Not as easy as with animal cells. Okay, so this is animal cell, plant cell. We also have, of course, fungal cells. So fungal cells. And fungal cells are pretty similar to the animal cells. Like animal cells, they don't contain chloroplasts. They might contain, so no chloroplasts. They, of course, contain nucleus. They contain mitochondria. They contain endoplasmic reticulum, Golgi apparatus, and things like that. Membrane-bound compartments. And they also have very often a cell wall. But this cell wall is not necessarily made up of cellulose, like a plant cell wall. This is usually made up of a slightly different substance called chitin, which is sugars and also amino containing, nitrogen containing compounds. But otherwise, fungal cells are also eukaryotic cells, which contain these internal membrane-bound organelles and a proper nucleus. Now that brings us back to the nucleus, and the question is, we said with the red blood cells that red blood cells don't have, in mammals at least, don't have a nucleus. Do all cells, other than the red blood cells, do all eukaryotic cells have just one nucleus? So, or do they have? Can they have more than one nucleus? And the answer is eukaryotic cells can have more than one nucleus. So, for example, some skeletal muscle cells. They usually have several nuclei. Some protists, these are single cell, very small eukaryotic cells, and there are lots of protist cells. They are just a single cell, not a whole complex organism, and they very often have a micro and a macronucleus. Micro and macronucleus, I should say. And they have different functions, and very often in special fungi, we also see that they have two nuclei, which is referred to as a dikaryon, because they have two nuclei in there. So these are all the characteristics of typical eukaryotic cells. It's very important that we recognize that animal, plant, and fungal cells all have more or less very similar structures. They contain these endomembrane organelles, which allow the cell to carry out specialized functions in these different areas of the cell. So I hope this makes sense, and thank you very much for watching.